To kick things off, can you tell us a little bit about yourself, Fay Wei? What some of your background and your research that you do?
Yeah, so I grew up in Taiwan, which is a tropical, subtropical island. There are a lot of really cool stuff over there, and my parents has a little cabin in the woods, and we would go there almost every weekend, And this was a day before smartphone, so the only entertainments you had is either books or going outside, and I particularly like field guides because they're more pictures than text, and it become quite fun to try to match what you find in the fields and in the field guide, and that's in part how I got into biology. So then I went to National Taiwan University to study biology, and I joined a research lab when I was an undergrad. And actually, I published 3 first author papers when I was in undergrad, and one of them
Really?
actually got cited 100 times so far.
Wow, as an
So
undergrad?
not too bad.
That's amazing.
And but then in Taiwan, if you're a healthy male, you have to go to military for a mandatory service. So yeah, I joined the army for 1 year, and I was for some reasons I become a tank commander.
That's awesome.
Yeah.
But you
Well,
didn't just drive a tank, you like commanded multiple tanks, right? Was that
yeah, four to be exact. Yeah, it's a really different period of my. I was an undergrad, right? It's totally free, and I can do whatever I want. And then you join the army, and
it's not like that at all, right?
everything is opposite. But then I started to apply for graduate school when I was in the army, and I went to Duke University to study ferns because there were a lot of ferns in Taiwan. I became very interested in ferns when I was young, and yeah, I went to Duke University to study ferns and did a quick postdoc at Berkeley and another one at University of Zurich before joining here.
Mm-hmm. So you had one paper at Duke, I recall, that made a pretty good splash. Got you in Rolling Stone, right? Can you tell us about that?
Yeah. Speaking of which, let me share my screen.
Can I call you Tank Commander from now on instead of Doctor?
Nope.
Right. I'll stick with Fay Wei.
Yeah, so when I was in grad school, my advisor and I, we discovered a new fern genus. And when you discover something new, you got a chance to name it, right? And we decided to name it after Lady Gaga. So the fern genus is called Gaga. And there are 2 new species that we describe. One is Gaga germinata. If you are familiar with Lady Gaga, her real name is Stephanie germinata. So this is for her family. And Gaga monstra parva, which means little monster in Latin. Again, Lady Gaga's fan call themselves Little Monsters, so this is dedicated to Gaga's fan, I guess.
Got a kind of a Halloween thing.
Yeah, it's true.
Little monsters.
But we didn't just do this for whatever. There was a reason, right? So ferns. This is a fern gametophyte on the right, and Lady Gaga once dressed up like a fern gametophyte in her Grammys performance. So we think it's appropriate. And there was a stretch of DNA in all the Gaga species that spells Gaga, and whereas the closely related genera spell something else. So it's encoded is in DNA. Yeah, like you said, we made it to a Rolling Stone. I've never imagined that as a fern biologist, who cares? Who cares about us? But we're on the Rolling Stones, and this is Lady Gaga's selfie actually with the Gaga ferns.
That's with the fern?
Instagram, and yeah, it's pretty cool.
That's great. Okay.
Yeah, so this is part of what we do. We are evolutionary biologists. We like to explore and discover and describe new things. So here at BTI, we have done a few things so far. This we find a new genus of green algae around Ithaca. We haven't decided on the names yet. We just call them Ithacan for now. We also discover new fern species from Madagascar and other new genus of cyanobacteria from Panama. Yeah, so this is part of what we do. But our newfound obsessions, at least to me, is homeworts. So homeworts are a group of bryophytes. No one really have studied them very extensively, but they have really interesting biology. So for example, most of them have carbon concentrating mechanism. So this is a way to enrich CO2 in their chloroplasts to enable a better photosynthesis. So this is a unique way to get carbon. And homeworts also have a rather unconventional way to get nitrogen source. So they engage in symbiosis with nitrogen-fixing cyanobacteria. So they have the specialized slime cavities right here, and the cyanobacteria come into the slime cavities and start to fix nitrogen for the plants, and plants will provide sugars in return. So they have these interactions. So yeah, we're interested in the biology of this. We using a combination of genomic tools, phylogenetic tools and genetics to understand how make these interactions possible, and how do they build this special carbon concentrated mechanism.
And they're slimy and they have warts, right? So they're so it's good for Halloween.
Yeah, I haven't thought of that. Yeah, they have like horns.
Yeah, I see the horns. Okay, well, before we get too far, let's talk about your first spooky plant that you're going to tell us about today. Something down in Australia, I believe.
Yeah, so start this. So I guess most of the stories I'm going to tell today, we do have some research connection to it. So I will first talk about the story, the plants, and then I'll talk about what we have been doing to related to that plant. Yeah, so the first one I want to talk about is in Australia. Australia is fairly big, almost the size of the United States. But for a long time, people don't know what's in the interior, right? At least for the Western world. So in 1860, the Australian government commissions these two persons, Burke and Wills, to plan expeditions to cross the continent for the first time. So the plan was they were started from Melbourne and go all the way up to the north to reach to the Gulf of Carpentaria and then return. And this will make the first cross of the entire continent. It was pretty ambitious. And on August 20, a total of 19 men and 50 horses started out. It was a big party and stuff. But it was a really difficult journey. After 2,000 miles and so on, after 6 months, they finally made it to the north. But on the way back, they encountered some problem. They ran out of foods, and people died. The horses died, of course, and there were only three people left. So Wells, Burke, and another officer called John King. So they were running out of food, and they noticed there's this cool little fern that they sometimes see the native Australians would collect them and eat them. So this is what they call nodule. Marsilea is a genus name. Now they don't look like your regular ferns, right? But they're actually ferns. They are not clovers. They are not oxalis, but they are true ferns. So the native Australians, they didn't eat the leaf, but instead they eat this little pots down below, right? So what they would do is they will collect these pots, these nodule pots. They will smash them against stones, and they will add some water and bake them over open fire to make nodule cakes. And so these three gentlemen, they find a big patch of nodule ferns, and they started to have a great feast on them. And Wells wrote in his diary that it seems to give me greatest satisfaction. So they were satisfied, they were full, and so on. But then they kept eating this. A few days later, he started to notice something went wrong. It seems to give us no nutrients. So no matter how much they eat, they still feel really, really weak and really difficult to move. And it become really, really bad so later. And one night, whales ate his last nodule ferns and never wake up. And soon, Burke also died as well. And John King, the third officer, was later rescued by the Aborigines and survived. So what went wrong, right? What's in this fern that make them, so full but yet so weak? And the later scientists found out they have tons of faminase. So this is an enzyme that breaks down vitamin B. So the more you eat nodule ferns, the fewer vitamin in your body, right? And that is making them full but yet weak, and then they die of malnutrition. But then you might ask, the Aborigines seems to eat this all the time, right? Why they are totally fine? So one hypothesis that the historians and the scientists have been thinking about is perhaps these three Western gentlemen, they grind up the nodules and they just add water and eat them as a soup or eat them as a porridge. Whereas the Aborigines, they bake them over open fire, and the heat would deactivate the enzymes, right? So sort of a detoxifying way by heating them, turn them into a cake. And this might make them, being able to survive versus die.
Yeah, that's important.
Yeah.
So it's curious. So these white explorers, they knew the Aborigines ate them, but did they not know that they made them into bread? Did they not pay attention? Did they just not care, decide to make soup instead? Do we know?
I don't know. They may have their personal preferences, maybe, or it could be just way easier to just eat them as a soup, as porridge.
Yeah, so you don't have to grind them up and stuff into a powder and make a paste. Yeah, that makes sense.
Yeah, so one thing I haven't really talked about is what are these pods, right? So these are actually the reproductive structures of this particular group of ferns. They are called sporocarps. So if you pick this pot and put them in water, and this is what will happen. Oops. So they will germinate. This is not a germination in a sense that the seed germinates, but you can see they push out this weird, again, spooky kind of things, right? And you can see all these yellowish grape-like things. Those are the sporangia. So the sporangia will contain the spores, and the spores will later produce either egg or sperm. And the way the spores produce sperm is really unique. So the whole process from spores, from rehydrations to releasing the sperms only takes about 8 hours. So this is quite rapid process. You can think about this. The cellular differentiation has to be pretty elaborate to go from spores to sperms. And to now there was no mRNA transcriptions. And all this process was done by splicing out introns so that the protein translations can happen. And this is quite a unique phenomenon. And right now, Nassim in my lab, she has finished the genome sequencing of Marsilea, and we have RNA sequencing data in different time points. So we can characterize at which stage, which class of mRNA got spliced out. So it's an interesting side project we have now.
Yeah, so just to clarify, normally you have your genes and then they get transcribed into RNA, right?
Right.
Then pieces of the RNA will get chopped out and spliced together, and then that spliced piece will be used to create protein.
Exactly, yes.
So these things, like, what do? They kind of store the RNA, they do the splicing in advance and store it so it's all ready to go.
So they make the mRNA, but they didn't splice out the introns. So
Okay.
for mRNA to make proteins, you have to splice out all the introns. So to make the mRNA, we call them pre-mRNA, that still have the introns in it, and they store those pre-mRNAs. And in certain times, a group of mRNA will get spliced out, and they will make a specific class of proteins.
I see.
So this is a quite interesting way to control development.
It is.
Especially in rapid development.
Yeah, I guess it would be beneficial evolutionarily because you could get your sperms out faster than the competition, right? And you
and this group of ferns, they are somewhat aquatic, but you can imagine, for example, in Australia, the interior, you only have the water is not everywhere, right? You have little ponds here and there, and the ponds will dry out because of seasonalities. So you have the response to the environments really quickly.
That's cool. That makes sense. That's really neat.
Yeah. So this is a story of eating ferns can kill you.
There are others are edible, so you got to be careful which ones you eat, right? So.
Yeah, and not all ferns. So here in upstate New York, we eat fiddlehead ferns, right? In the spring, we pick them, and those are fine. Those are good. Those are ostrich ferns that you can pick here. But in other parts of the world, for example, in Japan and sometimes in California as well, they will pick a different kind of ferns called bracken ferns. And the bracken ferns is notorious for causing cancers. Again, there's a special ways you can treat the fiddleheads to remove the cancer-causing stuff, but be careful.
Yeah. Yeah, and those they give like if cows eat them, they get stomach cancer and stuff too, right? Isn't that
Yeah.
bracken ferns often. You have another story closer to home too, right? Wolf's bane. Can you tell us about?
Right.
Such a pretty flower.
This is pretty flower. This is aconitum. Like you said, it's wolfspan, or sometimes called monkshood because it has that cool thing. Again, maybe spooky if you imagine this is, I don't know, black rat color.
Like the Grim Reaper, the purple Grim Reaper.
Right. So aconitum, there's a thing in aconitum, especially in the roots, called aconitin, that is highly toxic. It will bind to the ion channels in your neurons and really mess up your neural systems, and supposedly could cause really painful death. So in a lot of cultures, they will rub their arrows in the aconitum roots so that they can make a more efficient kills. There's also a story, well, I guess a horrific murder story that happens in 2009 in London. So this person, like Chima, so one night he ate a bowl of curry, and later it was found dead. So the way they figure out what happened is they did a lot of analytical chemistry, and they found, there's aconitum powders in his curry, and was later found that his ex-lover actually sneaking into his apartment and sprinkle the aconitum powders on his bowl of curry on the pot, and then because jealousy, I guess. So yeah.
And that was just 2009. That's
2009. Yeah. And the murder report was quite fascinating because it involved ethnobotany, of course, and some ancient Indian literatures that because they are of Indian origin, so there's something there as well.
Mm-hmm. So this plant, it's not just in upstate New York, right? It's pretty much exists.
It's everywhere, and it's a common horticulture stuff. So sometimes a lot of people keep them in their gardens. So people should wear gloves, of course, when you work with this. Otherwise, you might feel a little bit tingling because tiny amounts can pass through your skins and give you a little weird sensations when you work too much with them.
Yeah. Oh, yeah.
Don't eat them, especially don't eat their roots. Yeah.
Yeah. I won't. Thank you for that warning, Fay Wei.
But we have a little studies on the research project on Aconitum. We are not interested in the toxicity per se, but we're interested in the conservations of a particular species called Aconitum noveboracense. So noveboracense means New York in Latin. So this is again very close to home. The common name is northern blue monkshood. So in New York State, they're only present in a few places in the Catskills, usually along a few ravines. In addition to New York, so we're here, right? There are a few odd isolated populations in Wisconsin, Iowa, and Ohio. Part of what we're interested in here is, with the support from the Triad Foundations, we're trying to better understand these species in the New York State and look at their genetics and genomics and see can we better preserve them or not. So this is Susie Strickler, my colleague at BTI. She was surveying the Aconitum plants in the Catskills.
I think she's watching today too. Hi, Susie.
Yeah. Hi, Susie. So one big question we want to get to is this a distinct species or just a local variant? And this is because there has been a the species is quite confused taxonomically and for many years. Because some people think, so the reds, so these are the Aconitum noveboracense. Some people think they probably belong to a bigger species called Columbinianum in the West. Or some people think they might be part of the Untitled in green in the South. So they might not be a distinct species. And the implication of this is huge, right? Because if it's part of the species all in the West, probably you can make a case it's not too important to conserve them because it's part of the larger, bigger species, right? So this is what we want to figure out, and we generate some genomic data. And again, this is supported by Triad Foundations and NSF BTS summer internship programs. So Connor was an intern, oh my God, last year? I lost track of time. But so he was here and he looked into the genomic data we have generated. And it's pretty clear that noveboracense, the New York Aconitum, is a very distinct species, definitely not part of Untitled in the South, nor Columbinianum in the West. And so again, this is an important piece of information. It does argue that we really need to put more conservation efforts into this species because it's unique, it's distinct, it's not part of anybody else.
That's important. That's a nice initiative, and it's nice. It's a nice program. The undergrad summer intern program we have at BTI is really great for folks who aren't familiar with that. Maybe we'll do a breaking ground on that come next spring or so. That could be a good one.
Yeah.
Yeah, it's a nice project. So speaking of the intern projects, your next story also involves a summer intern, right? From last summer, 2019.
Nice transitions. So here we're in Ithaca, and we're proud of the Finger Lakes that in this area. But sometimes in the summer, something spooky will happen. You'll have this spooky green stuff on the lake, and these are the algae blooms, harmful algae blooms. And when this happens, you will often see signs like this saying the beach is closed. You should not go near the water because this algae, mostly cyanobacteria, will produce toxins and can be really dangerous to human and also your pets like dogs and so on. So It's a serious problem here, not just here, but at least close to home, I guess. So again, harmful algae blooms here, at least in this area, are caused by cyanobacteria, like microcystis, dolichospermum, and so on.
All four of those things, those are all cyanobacteria?
These are all different kind of cyanobacteria.
Okay.
Right. So you can imagine cyanobacteria never exist by themselves, right? So there's always a community of stuff, community of microbes that are associated with cyanobacteria. And you can imagine a scenario, maybe if you see this type of microbe presence in a water sample, it might give you some indications of whether the bloom might happen in the next few days or not, right? So this is something you want to test, want to explore more further. Then last summer, I believe, Victor was the summer intern in my lab, and we were thinking about the potential project that he could work on. And then the harmful algae bloom happened. So we kind of think, there might be something we can do here. So we went to the Taganic State Park and we got some water samples. So you can see the greenish stuff on the surface. Those are the cyanobacteria bloom. Yeah, so Victor took some water sample and we bring it back to the lab, brought it back to lab, and we pushed them through this really cute device called Nanopore MinION sequencer. So this is a DNA sequencers. I don't have here with me, unfortunately, but like this big. And what you do is you insert this thing called flow cells. It's a disposable thing. Insert it into the device, and you load your DNA samples directly into the flow cells, and you're plugging into a computer, a laptop, and it will start sequencing right away, and will give you sequencing results right away. And those are good DNA sequencing results. And so imagine if you talk about DNA sequencing, you probably will imagine humongous machines tucked away in a big facility elsewhere, and might take days to get the results back. But this, you can get the result back in real time. And it's super portable.
Do you need to isolate the DNA first, or can you just put your sample right in there?
You need to isolate your DNA first. Yeah. So in a way, this is like Spock's tricorder, right? Kind of a magical device. But again, I think there's a possibility that if you can figure out this process better, before you go for a swim in the lake, you can get your samples, get DNA somehow, and then sequence them on this device. And that will tell you what are the cyanobacteria inside the water, how many of them are present, do they have toxic genes or not? And that will maybe give you a green light, you can swim, or red light, maybe you probably should avoid that water. And this can give you results in real time. So it could be quite interesting. So the way
I was just saying that's great. That sounds like it would be very valuable.
Yeah.
About their dog, you could bring your dog to the park and not have to worry in case it gets in the water and starts swimming around and stuff.
Yeah. So the way I think about this is, imagine you have a bag of unknown chips. I have no idea what's in it, but it has a barcode, right? So you took your barcode reader, and that will tell you, what's in it. What's the nutritional values? Do they contain allergens or not, right? And in this case, this bag of chip could be a water sample, or the DNA derived from a water sample, and you take your sequencer as a barcode, and it will tell you a lot of information that you will need. So we are trying to develop this protocols better and to characterize what we can find, what we find so far. So this is still an ongoing project.
And this was also funded by Triad, is that correct?
The summer internship was funded by Triad, and we're looking for funding opportunities to support this part of the research. We are still in the very early stage, so we're hoping if we can get more preliminary results, maybe we can apply for larger funding source. But we're not ready for that bigger federal grants yet. We still need to collect a lot of data on this front.
Yeah, That's great.
Yeah, so Victor, the data that Victor generated, we're in the process of publishing it. So yeah, it's fun.
Okay. Okay, great. We're at 12:37. Normally I would try to wrap up and start the Q&A now. I don't see any questions yet. If people have questions, feel free to throw them in there. If not, we do have one more story prepared. So we could maybe I don't see any questions yet. So
Right.
you want to just tell your last story?
The last story, yes, of course.
Right.
So the last story, so okay, here is the plant tree of life, and I want to highlight put your attentions to this liverworts. So there's a group of bryophytes. They don't have vasculatures, like usually fairly small. And some liverworts can make you high, high in a way like the cannabis high. So Radula is a genus of liverworts that are known to produce parotinine, which is structurally similar to cannabinoids, the THC. And there's some experiments on mice that had very similar effects on to THC as THC. And people actually smoke this. So this is, if you go online and you can find a random quotes, this one in particular, like, I've been smoking a lot of this lately. I'm lucky that it grows in my backyard. So I basically have a small bag of supplies. I've literally stayed down, I'm so stoned.
That sounds like a bad idea.
Yeah.
It's just,
smoking random stuff. Right. It's a bit fishy because, so you go on eBay, for example, you can buy this thing. So this person in Australia is selling Radula marginata, real exotic moss seeds. There's so many problems with this sentence. So first, this is not a moss, right? This is a liverwort, and bryophytes never produce seeds; they produce spores. So from the description, it looks really suspicious. But my colleague George Gender, he's curious enough. He actually ordered this from this Australian guy, and indeed we got some random seeds back. So it's definitely a fake thing. Again, if you do more Google search, you also find this. They are selling Radula herbal extract, and it's labeled GMO free and organic. But again, George, my colleague, he ordered this online, and he did a mass spec to see what's in it, and of course he couldn't find any trace of parotinine or THC. So this is another fake stuff. What is not fake, however, is, I don't know if you can see this. I have two Radula growing here. I haven't smoked it, but it's sitting on my desk. Well, with again the support from Triad Foundations, we're interested. So this is showing the Radula plants I have in my lab. This guy. So we're interested in understanding how they make cannabinoids. So this is a collaboration with George. It was George's idea actually, and then Frank Schroeder. So yeah, we'll be combining genomic data, metabolomic data to understand how do they converge until they evolve the ability to produce cannabinoids.
So it's probably completely different from cannabis, presumably, because they're so far evolutionarily apart. They could be completely different pathways.
That's one possibilities, but oftentimes plants can fine tune a few enzymes that just recruit the existing pathways and just fine tune that a bit. They can make a quite different compound. So to be determined, still early phases. So we don't have any data to show, but it's a cool project.
Could be some good pharmaceuticals potentially coming out of that for.
Yeah, hopefully. I don't know.
Yeah, that's cool. That's another good one. Okay, we do have a question, and it's going back to the algal blooms. Could you please elaborate on some of the toxic cyanobacteria? So I don't know, maybe talk about the species, you know, what other, like what bacteria do they live with? It sounds like some cyanobacteria do well with some algae and other species do, you know, live with other species of algae or something. Is that right?
Yeah, so the most major cyanobacteria that produce toxin is microcystis. They produce microcystin, and that is a chemical that will produce will give damage on your livers. So it's pretty bad. The water sample we got, that Victor got, actually didn't have microcystis. It has mostly dolichospermum. And we couldn't find any microcystin producing genes. So in a sense, maybe that water sample isn't that toxic, isn't that harmful. And we do find a lot of other microbes associated with that water samples with dolichospermum, that's the cyanobacteria. But the problem now we have is we only have one water sample. So we're hoping we can generate more so we can start to do some comparisons. And we're especially hoping we can get more samples that actually has the microcystis, the toxin producing cyanobacterium. So that again, we need more samples to do comparisons to draw conclusions from.
Yeah. It was interesting. Well, there's many interesting things to me about this project, but one is this concept of metagenomics, where you're not taking one species and trying to figure out the genome, which is what we often hear about when we hear about genomics, but you're just taking a water sample and whatever's in there, you're sequencing everything that's in there, all the genes that are in there. So the genomes of who knows how many species. And
yeah, we also, in that genome, in that water sample, there are quite a few interesting things. We found several invasive aquatic organisms in the water sample as well. So they may have, left their DNA elsewhere, and we're able to pick them up in our water sample, in our samples. There's also a few human DNA there.
be careful there.
Yeah, so before the approach is usually more targeted. You target a certain regions of the genomes and you sequence that regions. And our approach is just sequence whatever is present. And Nanopore, the really good advantage is they give us really long reads. With long reads, it's easier to piece individual sequences back together, back into a longer context of genomes. Right. So that give us more information and can characterize the metagenomes better.
Yeah, the way I think of that is like if you have a jigsaw puzzle.
Yeah.
A foot by a foot, if it's 1,000 pieces, that's hard to put together. If it's 25 pieces, it's a lot easier to put together. So the longer reads, it's like fewer pieces to a puzzle. Does that make sense?
Yes, that's exactly right.
Okay. Any more questions from folks out there? We've got Fay Wei, are you do you have any good Halloween costumes? Are you dressing up this year? You do anything for Halloween? Dress up as a toxic fern? Are you gonna dress up as a toxic fern or anything?
Yeah, I am not a big fan of Halloweens. I'm sorry.
Okay. Oh, we just got two more questions. One is, are you most interested in studying plants for their potential in medicines, toxins, or nutritional compounds?
Me, not particularly. I like you said earlier, I like weird biology. So any plants or any organism doing weird stuff, I'm interested in studying them. Maybe some of them might have some potential applications. So for example, our newest obsessions like homeworts, right? It has cyanobacteria symbiosis for nitrogen fixations that might have some long-term applications if we can figure out the mechanisms. There are carbon concentration mechanisms. Again, it's an interesting phenomenon that can boost photosynthesis and can boost yield. So if when we figure out how to make that, maybe we can put it in a corns or in a rice. Maybe that will be more long-term applications.
Yeah. And here's a related question from Armando. Hello, Armando. What's the most important known benefit of hornworts in general to ecosystems? And what sets them apart from other plants?
Wow. Okay. So hornworts are a very small group of plants. There are only 200 or so species. You don't, if you pay attention to them, you can find them. But a lot of people haven't never seen them, even botanists. What's the benefits to the ecosystem? Man, I don't know. Yeah, I don't know if anyone eats them. Yeah, it's a good question. Yeah.
We'll think about it and we'll have you on for part 2.
Right, yeah.
Yeah, have some time to here's a really good question that actually I thought of and I forgot to ask. I'm just going to read the whole comment because it's really good. Your comments about the questionable marketing of liverwort definitely gives me pause about purchasing some homeopathic remedies. I don't typically, but they are widely promoted here in Mexico and even sold in most pharmacies. Is there any kind of broad advice you'd give people who might see a quote unquote natural remedy given the potential of some of these plant capacities and aren't sure how to evaluate it?
Yeah, the regulations again needs to be there, right? And with this has been a big problem. For example, in traditional Chinese medicines, we rely, at least where I come from, you go to the our pharmacy stores and you buy a bunch of dry stuff, right? And then you have to rely on the person who sells you so this like they are the correct things. But now with the DNA sequencing technology, you can easily sequence them, either some markers or just the whole genomes, and you should be able to tell if they are what they say they are. So there's for example, a really good case. I think it's a tuna in New York City. So they sample a bunch of tunas from a bunch of different sushi stores, and they sequence it and think most of the time they are not what they labeled. So they are totally different species than they are selling.
Really?
Yeah. So to what extent the regulations apply to this, I'm not the expert, but yeah, it's definitely could be an issue.
Yeah, and you say it's really easy to do the sequencing, and I get it's probably easy for you to do the sequencing.
That's true. That's true.
You know, I don't know if it'd be easy for me to do the sequencing. I mean, I remember when I was an undergrad and I worked in a lab for a year and I was isolating DNA, and I don't remember how to do that. And then, and I actually had that experience. So, you know, I don't think my mom could, or, you know, those folks, but it is cheaper now. So maybe you could send it away to somebody else or have somebody else do it for pretty cheap now with these nanopore things. Maybe.
Yeah, the rumor is that the company is making iPhone versions of it. So
Wild. That is wild.
so yeah, maybe we'll have the true tricorder soon.
That'd be amazing. Okay, one more question. If you could pick any fern or seed-free species to sequence the genome of next, which would you choose?
Oh boy, I would choose a fern called Ophioglossum. So Ophioglossum is a
Phenoglossum?
Ophioglossum. So the common name is adder's tongue. The cool thing about this fern is they have the highest number of chromosomes ever known. So they have 1,400 chromosomes. So like human, you have like, right? But they have 1,400.
We have what, 23? Like
yeah, 46 if you double them.
46,
but
yeah.
So I would definitely want to sequence it and see what's going on there.
That's wild.
1,400. How do you do mitosis? There's a lot of chromosomes to parse to pull away and to make it correctly, right?
Is it haploid Are most ferns haploid or no? Am I misremembering?
So this particular ferns are probably a polyploid or recent polyploid. That's why they have this number, this big number of chromosomes.
It just keeps doubling and doubling and doubling and doubling until you have this some gigantic number.
Yeah.
That's wild.
Yeah.
Very cool. Okay. I don't see any more questions. Oh, we just got one more. Are you and Ruth Richardson planning to work together on early detection of microcystis in the lake? I'm not sure who that is.
Yes, yeah. We've been talking a lot with Ruth, and we applied for several internal grants together. Yeah, she's amazing.
Is
We have
she
complementary skills and complementary interests. So yeah, it's perfect.
is she a professor at Cornell or?
Yeah, she's a professor at Cornell who does like this person said early detections. So she has a different set of system that can see if certain toxins are present, so that certain toxin genes are present or not. And they have done way more samplings than what we did. We're just a beginner, and they have done a lot of really nice foundations on this area. So yeah, we're definitely working together with them.
And that was from Lynn Leopold, asked that question. All right, we're at 12:53. I see no more questions. I think this was a really good one. I love to see this engagement from the audience. That was a lot of fun. There's lots of good questions. Okay, I think we'll wrap up here with no more questions. Thank you, Dr. Fay Wei Li, for taking the time to chat with us. And that was a lot of fun stuff. I really enjoyed this one. And thank you again to everyone out there in the audience for joining us and engaging. That's what makes this so great is getting that engagement with folks out in the community. And I'll just let you know, you can read more about BTI's current research and many other neat stories. I don't know if there's much spooky stuff, but last year's annual report, I'm trying to think if there's anything really spooky in there, I'm not sure. Might be. And that's at www.btiscience.org/annual report. And it's really, it's all digital. It was the first time we've done an all digital one, and it's really cool. There's, it's interactive. There's links to videos, all kinds of neat stuff in there. And it looks like Mike put the URL in the chat box, which is great. Thank you, Mike. And if you could please join us for BTI's next Breaking Ground discussion series, that will feature an interactive chat with BTI's Lucas Mueller on Wednesday, November 18th. So we're doing it before Thanksgiving, doing a little, usually these are like around the end of the month. The next one's gonna be in the middle of the month before American Thanksgiving. We'll be talking about using big data to help feed the world. So Lucas does some neat stuff with breeding databases with sweet potato and yams and banana, cassava or yucca. So we'll be talking about a lot of really great stuff that he's doing globally. That'll be a good one. Please don't miss it. And you can go to btiscience.org for more information and register. And just would like to remind everyone, BTI is an independent nonprofit research institute, and we operate in part thanks to an engaged audience like you folks and your generosity. And if you'd like to make a gift to support BTI, you can donate online at www.btiscience.org/give or email our development team at development@btiscience.org. And thanks again for your interest and support of the Boyce Thompson Institute. And have a wonderful day and be well.
Bye.
Bye. Oh, the same. Thanks again, Fay Wei.